The regulation of protein function is something we've talked a lot about. And one of the ways you can regulate a pathway is by compartmentalizing it or putting it in an organelle, such as the mitochondria. The only way you can have a pathway on is if the substrates are being brought into the mitochondria for energy production. In this lecture, we're going to start to look at the role of mitochondria for energy production using pyruvate from carbohydrate catabolism, and we'll also look at how the pyruvate dehydrogenase complex is regulated.
Mitochondria really are the hub for energy production, and we'll talk a bit about its structure and how it helps produce 90 to 95% of the energy in a cell. We're going to start by looking at the pyruvate dehydrogenase complex, or PDC for short, and it uses the pyruvate that we made in the last lecture in the breakdown of glucose in glycolysis. The pyruvate dehydrogenase complex is going to make a molecule called acetyl-CoA, and this will get used in the citric acid cycle, which I'm sure you've covered a billion times in your biology courses. But what we're really going to focus on is just how important it is as a central pathway for metabolism as well as energy production, and this will all take place in the next lecture. We'll look at the role cofactors play in the pyruvate dehydrogenase complex's mechanism of action. And as with all of the pathways, the regulation is going to be super important. So we'll see how the PDC can be regulated in order to allow for the cell to regulate what is happening in the mitochondria. These are the supplemental readings in Stryer available through the link on Quirkus. Eukaryotic cells are unique in that they have different organelles, including mitochondria, that can help compartmentalize different pathways and processes which allow for their regulation. Obviously, if you can't get a substrate into a cell or an organelle, it's not going to be used in that pathway and the pathway will essentially be off. On the left is an electron micrograph of a mitochondrion, where mitochondria is just the plural form for multiple mitochondria. And these can be found in most eukaryotic cells, with the exception being red blood cells. Now, it's been proposed that mitochondria actually have evolved from bacteria, which initially set up a symbiotic relationship with a larger host. So this just means that they've been incorporated into cells so that they might both benefit from each other. Mitochondria are similar in size to prokaryotic cells, and they also divide by binary fission. We can also see that they have their own DNA to produce mitochondrial proteins, and it was originally believed that we inherited our mitochondrial DNA from our mothers. Now, there are some cases of mitochondrial diseases that arise due to mutations in the maternally inherited mitochondrial DNA, resulting in mutated mitochondrial proteins. But we can also see that wild-type paternal mitochondrial DNA as well as wild-type proteins might exist. So it might just be that the paternal mitochondrial DNA is being eliminated. So there's still lots of research being done on mitochondria in order to obtain a complete understanding of their structure and function and how they've evolved. The mitochondria are obviously most well known as the powerhouse of the cell, and they can oxidize intermediates from the different complex macromolecular fuels in order to generate energy in oxidative phosphorylation. Now, this is all going to take place in the matrix or on the inside of this inner membrane, and many of the proteins that carry out these processes are either going to be soluble in the matrix or membrane bound. So mitochondria have this double membrane structure where the outer membrane contains porins or membrane proteins that allow molecules to cross, versus the inner membrane that also has transporters but is more selective in what gets across that inner membrane. The mitochondria have these structures of inner membrane called cristae, which are essentially folds or invaginations of the inner membrane that will actually create a greater surface area for these membrane proteins. This means that you can have more of them carrying out the function on the cristae, and we'll see that ATP synthase, the main membrane protein complex involved in ATP production, is found in the inner mitochondrial membrane. We saw that glycolysis can break down glucose in the cytoplasm to produce two pyruvate molecules, as well as two ATPs and two NADHs. Pyruvate can then be transported across the two membranes of the mitochondria, so first across the outer blue membrane, then across the inner mitochondrial membrane into the matrix, which is where the reactions are going to take place and what we're going to look at in this lecture. So a porin will first allow pyruvate to cross the outer membrane, and then a more specific mitochondrial pyruvate carrier will import it into the matrix. The pyruvate dehydrogenase complex, which is going to be the reaction we look at today, as well as 7 out of the 8 enzymes in the citric acid cycle will be soluble and found in the matrix. There's also going to be one enzyme that's going to be membrane bound, as well as those involved in the production of ATP from NADH and FADH2 in oxidative phosphorylation. So in this lecture, we'll break down the pyruvate dehydrogenase complex and how it produces acetyl-CoA, carbon dioxide, as well as the reduced cofactor NADH. So as I've mentioned before, the pyruvate dehydrogenase complex is a complex of multiple copies of the E1, E2, and E3 subunits. And these will come together to form this larger structure which is shown on the right. The cryo-electron micrograph shows you just how large they really are, particles that are approximately 40 nanometers in diameter when you have a ton of these subunits coming together. Now, the different subunits will require different cofactors for catalysis, and some of them are prosthetic groups, such as thiamine, lipoamide, and FAD, while coenzyme A and NAD+ are considered co-substrates, so they come into the reaction and are converted into products. These cofactors are also vitamin derivatives, where thiamine pyrophosphate is a vitamin B1 derivative needed by E1, and lipoamide is derived from lipoic acid, which is found in your dark leafy green vegetables, while coenzyme A will contain that B5 vitamin pantothenic acid. Those are both used by E2. And then the E3 subunit is going to use FAD and NAD+, which are derivatives of riboflavin and niacin, or vitamin B6. So this is why it's really important to have lots of vitamins in your diet so that they can assist with the various enzymes involved in energy production. This is the net reaction catalyzed by the pyruvate dehydrogenase complex. And you can see it is a dehydrogenase, so it does involve the reduction of NAD+ to NADH in the mitochondria. Since NAD+ is reduced to NADH, pyruvate is actually going to be oxidized to acetyl-CoA. So if you remember the structure of pyruvate and that it has 3 carbons, 2 of these carbons are going to be transferred as an acetyl group onto coenzyme A. You'll see that coenzyme A is either written as CoA or as CoA linked to a sulfur and a hydrogen, and it's this sulfur that will get used in order to activate the acetyl group in a thioester bond. The third carbon from pyruvate is going to be released as carbon dioxide, or CO2 for short. So this is the very first reaction we've seen so far that produces the waste product CO2. Now, I've also given you the net standard Gibbs free energy change for the reaction. So it's minus 33 kilojoules per mole. A very favorable reaction is written. Is there anything in this reaction that tells you why it is spontaneous in the forward direction? If you said the release of CO2, then you're right. It's kind of like when you open a can of pop, that psh, and release of CO2 results in an increase in entropy as we release this gas. So this reaction can actually be described as an oxidative decarboxylation reaction where we are making the reduced coenzyme NADH, and that's going to be important for energy production in the mitochondria. And we're also releasing carbon dioxide as a waste product. I just wanted to show you the structure of acetyl-CoA, where we have that acetyl group shown in pink attached to the sulfur from coenzyme A. The coenzyme A portion is quite a bit larger than that acetyl group that it's carrying, and it's going to be the E2 subunit that's going to transfer the acetyl group from pyruvate onto that SH group, creating this new thioester bond. Thioesters also have a large standard for energy change when they get hydrolyzed, meaning that if you use water to hydrolyze that thioester, 31 kilojoules per mole of free energy is going to be released. Okay, so this will produce acetate and a free coenzyme A. So essentially the PDC is creating this activated form of those two carbons coming from pyruvate, which is going to be important when we look at the reactions in the citric acid cycle. If we zoom in on the reaction mechanism from this large enzyme complex, there will be multiple copies of each of the three different subunits, and I'm only showing you one of each in different colors on this slide. And individually, they'll be able to catalyze their own reaction, but they will be dependent on the reactions that are catalyzed by the other subunits, so either before or after them, depending on when you're talking about the reaction mechanism. Okay, but by associated with one another, this is allowing us to funnel intermediates from one subunit to another. So on this slide, I'm just showing you the reaction mechanism where we're going to start on the left. So the E1 subunit, or pyruvate dehydrogenase, which is what the complex is essentially named after. It's first going to bind to our substrate pyruvate, and it's going to catalyze that decarboxylation step. Okay, so the acetyl group from pyruvate is going to be covalently attached to that thiamine pyrophosphate cofactor, and the carbon dioxide is going to be initially released. That E1 subunit is then going to pass the intermediate onto the SH group of E2. So that's just shown here as step number 2, where we're going to use that lipoamide cofactor that's linked to a lysine side chain in order to form an acetyl lipolysine intermediate. That acetyl group will then be transferred onto the sulfur found in coenzyme A, which is shown here as step number 3. Acetyl-CoA is going to be released from the complex, and then that last subunit, E3, is going to be needed in order to produce our reduced NADH and return the rest of the enzyme back to its starting state. So remember, one of the features of enzymes is that they're never changed in the reaction. So this is where that the FAD cofactor is going to come into play. It's going to ensure that the lipolysine prosthetic group is returned to its oxidized form. So there's going to be a series of redox reactions using the FAD cofactor, but the only reduced cofactor we make as a product is NADH. So FAD starts as FAD, it gets reduced to FADH2, and then oxidized back to FAD. Now, not shown on this slide are obviously all the other amino acids that can be found in the active sites of either the E1, E2, and E3 subunits, and these are going to help hold these substrates and intermediates in place to allow for the reactions to occur. So the pyruvate dehydrogenase complex really is the link between the breakdown of sugars in the cytoplasm and the production of energy in the mitochondria. It's found in the matrix of the mitochondria, and it is a soluble complex consisting of multiple copies of the E1, E2, and E3 subunits that'll use those 5 different cofactors in order to form the activated acetyl-CoA. The creation of this thioester bond is coupled to the oxidative decarboxylation reaction and will also make NADH in the process, which will then get used in oxidative phosphorylation. These reactions are highly favorable in the forward direction and are essentially irreversible, so that should tell you that regulation is going to be important. The reason why the pyruvate dehydrogenase complex is highly regulated is that acetyl-CoA can also come from the breakdown of fatty acid chains. So if we already have a lot of acetyl-CoA coming in from the breakdown of fats, this can slow down the pyruvate dehydrogenase complex and inhibit it. We can then slow down glycolysis and instead store any excess glucose as glycogen for use in muscle and liver cells. So whenever there is a buildup of products, there is going to be this feedback inhibition in the subunits that catalyze these reactions. So lots of acetyl-CoA that was either made by E2 or from the breakdown of fats can inhibit the complex, while a buildup of NADH can also inhibit at the E3 subunit. So this NADH can either come from the reaction itself or from reactions in the mitochondria. Now we've seen this trend before with hexokinase where the product of a reaction can inhibit the reaction. And these molecules can also have another influence on the regulatory mechanisms, which we'll look at next. The pyruvate dehydrogenase complex is also regulated by covalent modification. If you think of the complex as a bunch of grapes, there are regulatory enzymes that are part of this fruit salad. A kinase that adds a phosphate on to inactivate it, and a phosphatase that can remove the phosphate to reactivate it. Now, if you're going to put the phosphate on one of the types of subunits, which one would you phosphorylate? E1 is actually the subunit that gets inactivated through a conformational change when it's phosphorylated, and this makes sense. It is the first reaction to occur, so you would want to prevent it from reacting before the other two reactions take place. This can also allow the pyruvate to then go on and be used for other reactions. These regulatory enzymes, pyruvate dehydrogenase kinase and the phosphatase, both themselves also need to be regulated. This is where acetyl-CoA and NADH come into play again. They can both allosterically inhibit the complex, but they can also stimulate the kinase to phosphorylate E1 and provide a more permanent form of inhibition. So when your muscles are at rest and we don't need a lot of energy, these intermediates will start to build up because the CAC and oxidative phosphorylation will slow down, and they can then allosterically stimulate the pyruvate dehydrogenase kinase regulatory enzyme to turn off the complex. When you need a lot of energy, we see that the substrate pyruvate and ADP, which signals for low energy, will actually inhibit the kinase. This will prevent the phosphorylation and inactivation of the complex. So when the kinase is off, this will allow the phosphatase to take over and dephosphorylate the E1 subunits, reactivating the complex. The phosphatase can also be stimulated by calcium, which is really important for muscle contraction, say when you're running. And this will allow for the activation of the complex so that pyruvate can be converted to acetyl-CoA, and then that will get used in the citric acid cycle in order to produce reduced electron carriers for ATP production in oxidative phosphorylation. The pyruvate dehydrogenase complex can be regulated in a bunch of different ways. There's a kinase that can regulate its activity by phosphorylating E1 to inactivate it, while the phosphatase will restore its activity by hydrolyzing off that phosphate. Feedback inhibition can inhibit the E2 and E3 activity by the products acetyl-CoA and NADH, and these molecules can also allosterically stimulate the kinase to turn off the complex. The substrate pyruvate and the low-energy molecule ADP can also inhibit the kinase, allowing the phosphatase to dephosphorylate the complex and allow for energy production. And finally, calcium, which is an important signaling molecule, can also stimulate the phosphatase activity. And levels of calcium can be elevated when muscles are contracting when we need lots of energy. So the pyruvate dehydrogenase complex is an important step that takes place in the mitochondria, linking carbohydrate metabolism to the citric acid cycle. We first need to import the pyruvate into the mitochondrial matrix, and then the pyruvate dehydrogenase complex will catalyze an oxidative decarboxylation reaction producing acetyl-CoA, and this will contain that thioester bond. We first see that the production of that CO2 waste product is going to occur, and then eventually we'll also produce an NADH in the mitochondria for oxidative phosphorylation. The complex does contain 3 different subunits that carry out individual reactions, but they're all dependent on the presence of 5 vitamin-derived cofactors, some of which are covalently linked prosthetic groups, while coenzyme A and NAD+ are considered co-substrates. Because the net reaction releases a huge amount of free energy under standard conditions, it is essentially irreversible, so we do need a way of regulating it and controlling it, and this will all be done via the energy requirements of the cell. When we have the presence of different intermediates that can also get used to make ATP, this can also help with regulation. We see that allosteric regulation is occurring on both the subunits and the regulatory enzymes, and we also see that covalent phosphorylation of E1 inhibiting it. Make sure you understand just how important these cofactors are and review the different ways the complex is regulated before attempting the brain break questions on Quarkus.